Cécile, thank you for sitting down with me. Much appreciated. We are here at the Upov meeting of the Working Group for Biochemical and Molecular Techniques, the BMT. Do you have something similar at CPVO?
Yes, we do. We have a group that is called IMODUS. It stands for Integration of Molecular Data into DUS Testing. And IMODUS is a group that has a slightly different way of working that the Upov BMT group. The Upov BMT group is a place where all the states can come and share their experience in new molecular tools and approaches. And IMODUS is a group where CPVO selects experts and invite them specifically to talk about topics that we think are of interest for the European examination offices. So for us, Invite and the BMT are completely complementary.
Excellent, excellent. So what you mentioned IMODUS. What activities do they exactly do?
So IMODUS has been created 3 years ago now. So his objective is to implement the CPVO's R&D strategy. It works like a think tank where the experts have to reflect, discuss, assess the new molecular tools. They can propose R&D projects, and they're also integrated into the process of reviewing of all the R&D proposals that are involving molecular aspects. So the experts are all European, basically, but we can also ask non-European partners to come and share their experience on some specific topics. Among the European experts, you can find people representing breeders' associations, the examination offices, of course, universities, research institutes, and there's also some members of the commission from DG Santé or Agri.
We are here at the BMT discussing the use of molecular techniques in variety testing by DUS examination offices. In your view, should DUS examination offices be using more of such tools?
So for Upov members, the basic of the DUS testing are phenotypic descriptions, and BMTs are considered as complementary tools, but they are of great interest because they can increase the quality, the efficiency of the tests by reducing their cost or their duration. So it's very important. They can also be used to enforce plant breeders' rights. So the answer is yes. We support the development of molecular tools. We think that there should be even more. We have already a lot of examples of application according to the different Upov models. So for Upov Model 1, we can replace some assessments of difficult or cumbersome characteristics by molecular markers. So we have examples for disease resistances, for assessing also male sterility for vegetables and also for arable crops. And we have also examples for the management of the reference collections. So through Model 2, which is the combination of genetic and phenotypic distances for the selection of the varieties in the second growing cycle of the GUS testing, we have already systems that are implemented for maize, for lettuce, for barley.
Excellent.
And for what we would be called the Model 3 now, which is another approach that is based only on the use of molecular tools for the selection of the varieties in the first growing cycle. We have already a validation for French bean.
Okay.
So there will be there are a lot of other studies going on, and there will be more and more tools, and we're very happy about that.
Yeah. It's not so long ago that these techniques were introduced. And do you see a trend among examination offices? Is it being used more or less? Or
for Model 1, for the replacement of phenotypic assessment by molecular tools, there is no acceleration. It's more regular development of new markers.
Okay.
Based on markers that are published in the literature. So it's just going slowly but steadily. There is something new regarding Model 1. It has just been proposed to Upov. It's the use of markers that can help detect some genes that are involved in DUS traits, but without giving a complete information on the level of expression. So it's very new, and for this type of markers, so they can be very interesting, but you need to be very careful to have a really good understanding of the genetics behind the traits, and also to have very precise procedures of implementation to avoid taking a risk of having a wrong decision.
Mm-hmm.
So in case of DOP, there will always be a validation by biotest anyway.
Yeah.
So I see more a trend for the management of reference collections, because all offices have to face so the increase in size of their variety collections. So they really need to find means of reasoning the setting of their trials. So it's for that it's really important, and they invest a lot of efforts in interacting with each other, also to ensure the completeness of the variety collections and the precise identification of the varieties. So for that they develop, in some cases, some shared databases, which are very interesting tools. But sometimes they are a little difficult to develop because, for example, for maize, we supported the development of a maize database, a common maize database, but in the end it's not so common because when we compared the collections, we realized that for some countries, they had very few in common. So we had to develop two sub-databases for countries that were submitted to Western European conditions, let's say, and another one for the more Mediterranean conditions. So it's the other trend that I see is in the evolution of the molecular markers. So there's still a lot of applications where SSR markers are used. But for the new tools, it's more and more going to SNPs and also other kind of sequencing information like indels or even structural variants. So it's something that is going on. There are also other kinds of markers that are explored, like epigenetic markers. They're also promising. So we have a project that CPVO is financing for the distinction of mutants in apple. So the principle will be to study the level of DNA methylation in the whole genome of a set of mutants, and to try to detect epigenetic marks that could be correlated to the phenotypic changes that are observed in the mutants. So that's the main trends that I see.
Very interesting. I believe it was the Gala apple, the Gala variety that had many mutants.
Yes.
Interesting. I look forward to the outcome of that piece of research.
And I can see, just to add also, that examination offices are not really specialists so far of these new technologies of sequencing, but they are very interested, and they can. So usually they develop their markers using what's published in the literature, but they can also collaborate to research projects such as INVITE, which is the big European project on variety testing, where they will have the occasion to collaborate with scientists to develop new sets of markers by using whole genome sequencing for the fingerprinting or the genome-wide association studies for the DUS traits. So that's also the start of a trend. I think it will increase.
Interesting. Lots of different trends and further development of the various tools. But I also see that some countries, they are quite far ahead in the use of these tools, whereas some other countries, they hesitate a little bit. Is it necessary, and how could we bridge that gap?
To work with BMTs, I mean, you need really a high level of investment. You need, I mean, you need to make some efforts in terms of R&D developments, and so it's something that offices accept to do only if they're really sure to get a high benefit from this investment. And so usually it has a consequence that they work mainly on the big crops for this type of tools. I mean, it's very rare when they focus on more minor crops because of this level of investment, but it can happen. But even for this big crop, sometimes the offices with the smallest financial means will not have the capacity really to develop them. So knowing that, CPVO is trying to support the development of molecular tools through the activities of the Emojis group, but also by supporting research proposals that can be submitted every year. We have a call every year where offices associated to scientists or not can prepare some research proposals that we can co-finance.
Okay.
So usually this kind of projects, I mean, we strongly suggest that all the examination offices that are entrusted for the species at hand are participating to the project. So for the smallest ones, they're very interesting, but they cannot participate really in the development of the tools. So they're involved in the discussions all along the project. They can be involved in the validation, the implementation, but sometimes it can be difficult even with this help. For example, for the potato database that we supported also, we had small offices associated to it, and they have some difficulties to continue to participate because, I mean, to give you an example, the profiling, the cost of the profiling is as high as the examination fees. So it creates problems and it can be a limitation. So, I mean, to conclude on that, the investment that an office will put for this BMTs is really, I mean, in his hands. I mean, he has to take into account a lot of different parameters, and it's so when they hesitate, it's really because there are reasons behind the. It's not.
Yeah, sure. So you mentioned the cost as one of the main challenges for the use of such tools. Are there other challenges?
Yes, there are some challenges. It's not so easy to implement BMT tools. So for if I can structure my answer, I mean, if I think about the model one, so the replacement of phenotypic observation for, biomolecular tools, molecular markers, the main challenge is the knowledge you have about the traits. And even when you have a good one at a given moment, and I mean, enough to develop very efficient markers, they can very quickly turn a little less useful because of the evolution of the characteristic. And so you need to adapt, and it's one of the subjects that we are reflecting upon even in the framework of the Upov BMT group, to try to provide some ideas for the development of guidelines that would allow a continuous adaptation of markers for DUS traits.
So what other challenges exist?
So for the management of reference collections, the biggest challenge is to manage to define the thresholds that you need to apply the markers. And for that, you can ask experts to compare a couple of varieties with, distant from with a range of genetic distances, and then to try to associate their assessment to these genetic distances to see if there's a level of correlation. There is never a total correlation, of course, but you need a certain level to manage to define a threshold for genetic distances, for example. And so that's challenging because they're very specific experiments, and it's even more challenging if it implies several offices if you want to develop a common threshold, because then you have to set up in parallel different, I mean, the same trials, but in different environments. And also you need to harmonize the way the experts are making their assessment. In terms of challenging, as you mentioned, the cost. I mean, even if the cost of a molecular data point has been really reduced, if you want to invest in the technology in-house, it's still very expensive. I mean, to acquire all the machines and all the tools. And another challenge is also for examination offices to develop the expertise. I mean, whether you do it yourself or you subcontract, you have to have internally some expertise to know how to use efficiently the molecular data.
Obviously. And then of course the inter-lab calibration is another challenge.
Perhaps, in terms of harmonization. Yes.
Cécile, seeing all these developments, and I'm sure that the costs per data point and of the machinery will go down. Do you think that over time, variety testing will or perhaps should evolve towards full molecular testing?
So in a very, very distant future, we can imagine that it may switch to complete molecular. If the tools continue to develop as fast as they do, there will probably be some opportunities to get closer to this point. But for now, when we ask the experts, there is no consensus in deciding if this type of approaches would compromise the effectiveness of the protection. The risk, I mean, the concern is that you could use endless list of markers to manage to find differences. And so that would have to be framed by specific thresholds to develop for each crop, and it would be very difficult. So for now, it's so there have been some studies in the US about this possibility, but they were really not conclusive. So for now, we'll all agree that it's too early.
Still far away.
Yes.
Okay. Thank you so much, Cécile.
You're welcome.